Very good morning, students. Welcome to the e-lecture series, an initiative which has been taken by LJ Institute of Pharmacy. I would like to inform you that Pharma Ignite, it is an official YouTube channel started by LJ Institute of Pharmacy with an objective to provide e-learning to students in a current COVID-19 pandemic situation where the students they can follow the channel, they can learn from their homes, and they can be safe. Now, I, Dr. Jignesh Shah, working as a professor in the Department of Quality Assurance at LJ Institute of Pharmacy, would be teaching instrumental method of analysis in BPharm SEM7. In today's class, we will continue with the topic that is UV visible spectroscopy. Now, students, as you know that in the last class, we have started with the instrumentation of UV visible spectroscopy. We have covered different topics in the instrumentation. We have covered the source of radiation which are used in UV visible spectrophotometer. Also, we have gone with the collimating devices which are used in UV visible spectrophotometer. We have also gone with different types of monochromators which are used for converting a polychromatic light into a monochromatic light. In today's class, we will move forward towards another topic that is the sample holder or the cuvettes. Now, basically, whenever we are analyzing our sample, one thing which we should be keeping in mind in case of UV visible spectroscopy is in this particular technique, samples are analyzed in the liquid form. So if we are having a solid sample, then in that case, we have to dissolve it in a suitable solvent. And after that, it can be used in the form of a liquid sample and it can be analyzed. Whereas the liquid samples, they can be directly analyzed without dissolving in solvents and they can be analyzed by UV visible spectroscopy. Now for the analyzing a liquid sample, we need some sample holder so that that sample can be taken and it can be analyzed in the instrument. For this purpose, cuvettes are used. Now cuvettes, these are used for handling liquid samples. The cells may either be rectangular in shape or it may be cylindrical in nature. Now for study in the UV region, the cells, they are prepared from quartz or fused silica as a constructing material. So if we are working in UV region, that is from 200 nanometer to 400 nanometer, then we should use sample holders or cuvettes which are made from quartz or fused silica. And if we are working in the visible region, that is from 400 nanometer up to 800 nanometer, then the construction material for cuvettes, it should be used as fused glass. Now, quartz and fused silica, they are most commonly used in UV region, whereas fused glass, they are avoided in UV region because glass has an ability to absorb some radiation from UV spectra or the UV radiation region, whereas quartz and fused silica, they are completely transparent to the UV radiation. As a result, the sample holder will not absorb the radiation and the entire amount of the radiation will be available to the sample for the absorption process. Now, the holder or the sample holder or the cuvettes which we are using, they must be uniform in construction. The thickness must be constant throughout the entire holder. The material of the construction, it should be such that they should not react with the solvent, means they should be inert to the solvent. They also should not absorb the radiation, and that's why, as we have gone earlier, that quartz and fused silica, they do not absorb UV radiation, they are transparent to UV radiation, and that's why we are using this as the construction material in 200 nanometer to 400 nanometer analysis. The material which is used for the construction, it should be also transparent to the radiation. So these are some basic requirements for any sample holder to be used in UV visible spectroscopy. Now these are a few images of the cuvettes which are most commonly used in UV visible spectroscopy. Now out of this, the third cuvette is the most commonly used cuvette in case of UV visible spectroscopy in the labs. There are different other cuvettes with different sample size, mostly 10 ml sample size and 5 ml sample size cuvettes. They are most commonly used in the UV spectrophotometer instruments. Even 1 ml sample coverts, they are also used wherever the sample is very less in quantity, and this minimum amount of the sample it needs to be analyzed. In that condition, we use 1 ml sample covered for the process of analysis. Now, moving forward towards the another instrumentation, the component that is the detectors. Now, what are detectors basically? Now, detectors are considered as one of the most important component of the instruments of UV visible spectrophotometer, because these are the devices which converts light energy into electrical signals, and they are displayed on the readout devices. Means these are the devices which helps us to analyze that how much amount of the radiation has been absorbed by the sample and how much amount of the radiation has been transmitted. The final result of the detectors which will help us to analyze the concentration of the sample which is present. So these detectors, they are the last component of the instrumentation which helps us in quantitative analysis of the compound. Now these devices which are used for the analysis, they work on a basic principle that they try to determine either the absorbed radiation taken by the sample or the amount of radiation which has been transmitted by the sample. So basically detectors, they will analyze the transmitted light which passes from the sample and which falls on the detector. So whenever a radiation from source of radiation, when it is allowed to pass through the sample, some amount of the radiation will be absorbed by the sample and the remaining amount of the radiation will be passed towards the detector. So detector will receive only that much amount of the radiation which comes from the sample, and this detector will try to determine the intensity of the radiation which has been absorbed by the sample. So there are various different kinds of detectors which are used in the instrumentation of UV visible spectrophotometer. They are barrier layer cell, which is also known as a photovoltaic cell. The second one is phototubes. Also, they are known as photoemissive tubes. The third one is the photomultiplier tubes. They are basically the most commonly used detectors in UV visible spectrophotometer. So these are few detectors which can be used in case of UV visible spectrophotometer. As I have told, the detectors, they try to determine the intensity of the radiation which has been absorbed by the sample. So this is the basic principle which happens in case of detectors. And on the basis of the type of the sample which we need to analyze, there are different types of detectors which can be selected out of this photomultiplier tubes. These are the most commonly used detectors in UV visible spectrophotometer nowadays. Now, before we discuss these detectors individually in more detail, before that we'll go with the requirements of an ideal detector. So, first we should discuss that what should be an ideal requirement of a detector. Now, these are following requirements which a detector should have to be selected for UV visible spectrophotometer. That is, it should give a quantitative response. It means that the detector should be useful in determining the quantity of the sample by determining the intensity of the radiation which has been absorbed by the sample. That's why the detector should have a property to quantify the amount of radiation which has been absorbed by the sample, and hence it can have application in quantitative analysis. It should have high sensitivity and it should have very low noise level. So the sensitivity should be very high, because even a minor change in the sample, minor change in the concentration of the sample can also be analyzed by the detector. So such kind of detectors are most commonly used, which have high sensitivity towards a small change in the concentration of the sample or type of the sample. And even the detectors, they should have very low noise level, and that's why the signal should be highest and the noise should be lowest. That's why the quantitative analysis can be as precise as possible whenever the noise level it is very low. Now the detector should have a very short response time. So as soon as the sample passes from the sample and falls on the detector, within a very short interval of the time the result should be obtained. So they should have a very short response time. It should also provide signal or response quantitative to wide spectrum of the radiation received. Now, this is one specific quality of a detector, that the detector should provide the signal depending upon the wide spectrum of the radiation which has been received. Now, as we have discussed earlier, that UV visible spectrophotometers, they are mainly used from 200 nanometer to 800 nanometer. So this is the range of the radiation which we are using in case of instrumentation. So in this entire range of the instrumentation, the detector should have an ability to provide signal. Otherwise, the detector, it cannot be used in case of spectrophotometer. The detector should not be affected by minor variation in the temperature. As we have gone earlier, that the source of the radiation, they are associated with high temperature because we are heating the electrodes. And during the process of heating the electrodes, the temperature inside the instrument will go high. So these detectors, they should not be affected by this minor variation in temperature due to heating of electrodes in case of source of radiation. It should be able to detect lowest possible concentration. So as we have discussed earlier, that the sensitivity should be high. So lowest possible concentration of the sample should be analyzed by the detector. It should have minimum deviation from Beers and Lambert's. Means these detectors, they should follow Beers and Lambert's law, because the laws governing absorption of radiations are the Beers and Lambert's law, and that's why detectors should also follow Beers and Lambert's and should not show any deviation from Beers and Lambert's law. And the last ideal requirement of the detector is it should be robust in operation. So even a minor change in the operating parameters, it should not affect the performance of the detector. So these are some basic requirements of the detector. Now, starting with the first type of detector, that is a barrier layer cell, also it is known as photovoltaic cell. Now, it mainly consists of a metallic base plate. So in the diagram, we can see that there is a metal base plate which is either made up of iron or it is made up of aluminum. This acts as one electrode. On the surface of that, there is a thin layer of a semiconductor material or metal, which is selenium, that is deposited. And then on the surface of the selenium, it is covered by a very thin layer of silver or gold, which will be acting as a second collector tube or the second electrode. So this is the basic construction of barrier layer cell. So which consists of a base made up of metal, followed by a semiconductor metal known as selenium, and this is followed by deposition of another metal, that is a silver or gold. Now when light radiation, they fall on the selenium layer, electrons are generated at a selenium silver interface. As a result, whenever these electrons are generated, they become mobile and then they are taken up by the metal layer which is present at the base. Now, as a result, when electrons are transferred to the metal layer, a potential difference is created between both the electrodes, that is iron or aluminum as one electrode and silver or gold as another electrode. So due to this potential difference, a current is generated, and this current which is generated, it is been determined by the detector, that is barrier layer cell, which is connected to a galvanometer. Now, depending upon how much amount of the radiation that is falling on the surface of selenium, the current is generated. If higher amount or if high intensity of the radiation falls on the selenium layer, then more current will be generated. And if lower amount of the radiation falls on the selenium layer, then low radiation will be, or then low current will be generated. So basically, this is the functioning of barrier layer cell or the photovoltaic cell. So in the next class, we will continue with the remaining two detectors which are used in UV visible spectrophotometer. For more videos regarding other topics, I request you all to please follow and subscribe our YouTube channel, that is Pharma Ignite. Thank you very much.